Good afternoon. My name is Alžběta PUPÍKOVÁ. I'm part of the Lab of Molecular Mechanisms of Cancer, led by Dr. Zdeněk at the Faculty of Medicine, Masaryk University in Brno. As someone who is working in a cancer research, I often get asked a very fair question. How is it possible that after decades of studying cancer, so many people are still dying from it? And honestly, I completely understand that frustration because I share it too. The truth is, we have made remarkable progress in cancer treatment, and as a result, patients are living longer than ever before. But longevity doesn't always mean quality of life. Conventional treatments like chemotherapy often come with severe side effects, damaging not only cancer cells, but the healthy ones too, sometimes even increasing the risk of secondary cancers. So, while we can extend survival, the toll on the patient's body and well-being still remains immense. And that's exactly why targeted therapies were developed. They promise fewer adverse effects and a better quality of life compared to the non-specific approaches like chemotherapy. However, cancer isn't a single disease. It's not driven by one mutation or a pathogen that can simply be eliminated. It's a complex and evolving system that is constantly changing and even within the same tumor. And that's what makes truly precise targeting such an extraordinary challenge. And this phenomenon is what we call intratumoral heterogeneity. It's a mixture of distinct cell populations within a single tumor, for example, which is the intratumoral heterogeneity, or between different patients or different tumors. And we can also have a temporal heterogeneity, where we can see the change of the populations after treatment or just after time. And each population can behave differently. They can use different metabolic pathways, express different genes, and respond differently to treatment. And this diversity is exactly what makes cancer so difficult to treat. Today, intratumoral heterogeneity is considered one of the biggest obstacles to effective targeted therapy. And now I tell you how such a combination of targeted drugs may work. The first step in designing targeted cancer treatment is indeed a choice of targets. You can either inhibit an oncogene, which is a pro-cancer gene, or activate a tumor suppressor, which is an anti-cancer gene. And we chose the latter because boosting self-defense mechanisms of the cell may give us benefits in the long run. So what is tumor suppressor? These are specific genes that act as our body's natural defense system. They detect cellular damage, stop the cell cycle, and if necessary, they trigger apoptosis, which is for us the most important from the clinical view. And the tumor suppressor P53 is the most powerful of them all. It's a transcription factor, which means it binds directly to DNA and regulates the expression of hundreds of genes across the genome. But what makes it truly special is that it functions as a tetramer, four identical molecules binding together, which enables it to regulate gene expression with very high precision. Unlike most other transcription factors, it is precisely defined and evolutionary converse set of target genes. It's also active in nearly every tissue in the human body, which makes it one of the most essential safeguards against cancer development. But even P53 has its limits, and one of the main is its negative regulator, MDM2. In its inactive state, P53 is bound to MDM2, which marks it for degradation. However, once P53 is phosphorylated, it's released from MDM2 and can do some really nice things, like activation of genes that drive anti-cancer processes, which, as we talked, include stopping the cell cycle, triggering senescence, repairing DNA, and most importantly, inducing apoptosis. And since this entire tumor-suppressive cascade is controlled by a single master regulator, p53 represents an extremely attractive target for therapy. And when you look at this picture, naturally one may ask, so what if we target the MDM2 negative regulator to enhance p53 activation? And you're really not only one who asked that question. In fact, many researchers had the very same idea. What if we just simply inhibit MDM2 and let p53 do its job? And well, here's what happened. This is a list of MDM2 inhibitors that have already entered or are still ongoing in clinical trials. And they were all designed to do one thing, to make cancer cells go through apoptosis. But even their promising beginnings, none of them have made it to approval so far. Some failed due to toxicity, but most just simply didn't work as monotherapies. Why? Because instead of inducing apoptosis, they caused only a reversible cell cycle arrest, which is far from the true cell death we were hoping for. So why does this happen? Because p53 is not the only center of the cellular stress response. Cells have multiple safety systems, and activating just one is often not enough to push the cell into death. So, building on this idea, we ask a very simple question. What if we activate several stress responses at the same time? And that's exactly what we did. And this is behind the principle of our combined therapeutic strategy, which targets two parallel stress signaling pathways. The first is the p53 activation through MDM2 inhibition. And together with that one, we also focused on the integrated stress response. It's a conserved pathway that helps cells to adapt to different types of stress, like nutrient deprivation, oxidative stress, or DNA damage. When it's activated, it targets a key protein called AIU-alpha. When phosphorylated, it reduces the overall protein synthesis and translation at the same time. And selectively, it promotes translation of ATF4, which is another transcription factor that drives the expression of pro-apoptotic genes. And the synergy between these two pathways was clearly demonstrated in our recent experiments, as you can see here, with very strong apoptotic response in vitro and effective but not perfect response for the in vivo treatment. Because our goal is not only to stop tumor growth, but also to achieve tumor shrinkage. And so far, this has not been fully realized, and largely due to the high heterogeneity in treatment response. As you can see here, we use natlin and milademetan as MDM2 inhibitors and nelfinavar as AF2-alpha inhibitor. And in vitro, we got a really nice uniform activation leading to strong apoptosis. But in vivo, the response was a little bit weaker. Instead of apoptosis, we mainly observed reversible cell cycle arrest. And the different responses across the different populations is exactly the reason of tumoral heterogeneity. So, our main hypothesis was that when AIU-alpha isn't properly inhibited, the p53 network doesn't get strongly activated in some tumor cells populations. So, to say it easy, this means that these two pathways are in correlation. And what if, by combining therapies and adding one or two compounds, we could make those resistant populations finally respond and turn a partial effect into some real effective treatment. To test our hypothesis, we analyzed available single-cell RNA sequencing datasets from colorectal tumor samples. And now, before diving into results, let's first take a moment to understand what single-cell actually means and why is it so powerful for us. In a traditional bulk RNA sequencing methods, we average gene expression across millions of cells. You can imagine it like making a smoothie. You can taste the overall flavor, maybe recognize the strongest fruit, but you can't really tell what's inside. Single-cell sequencing, on the other hand, is more like a fruit salad. You can actually see every single piece. You can identify each cell, its unique gene expression profile, and how it differs from the rest. And that's what allows us to truly capture the full scale of intratumoral heterogeneity, to see how pathways like p53 and ATF4 behave across different cell populations within the same tumor. But we can take it even further. Because even when you know what each cell expresses, you're still missing one critical piece of information. Where are these cells located? And that's where the spatial transcriptomics comes in. You can imagine it like a map. It doesn't just tell you who the players are, but also where they sit and how the cells interact with one another. And only then you can start understanding the complex communication between cells that drives tumor behavior. Using this approach, we focused on the activity of p53 and ATF4, and we compared their z-scores, which basically means the activation levels, across different clusters of tumor cells. As you can see in this heat map, there was clear heterogeneity in the response in different clusters, because some clusters show strong activation of both pathways, while others were much lower. And just from this heat map, you can really see the correlation between the different clusters. We then confirmed this correlation also statistically with a correlation plot, and this shows us a significant positive correlation between p53 and ATF4 activity. And this was the key finding for us, because it not only supported our hypothesis and preliminary data, but also suggested that by targeting both pathways simultaneously, we really might be able to resensitize the previously resistant tumor populations. So, building on this, we used ingenuity pathway analysis, which is the bioinformatic tool that helps to identify key biological pathways and functions linked to our genes of interest. We paid particular attention to the non-responding clusters, which are those that show low activity of both p53 and ATF4. Because the pathways that are highly activated in these resistant populations could be the ones that represent the new potential for therapeutic targets. Altogether, these results provide strong support for our hypothesis and really open up new exciting directions for next step and potentially new therapy. However, this is still a proof of concept study, and we're actually just at the beginning of the story. Right now, we're expanding our analysis to include other cancer types because we want to compare heterogeneity and pathway activation across different tumors. We also obtained our own colorectal cancer samples from patients, and these samples will allow us to generate our own single-cell RNA-seq data. And we plan to combine it with spatial transcriptomics, both in patient samples and later in xenographed mouse models for in vivo validation. So that's everything from my side. A huge thank you to my supervisor and our amazing lab team. And thank you all for your attention and for giving me the chance to be a part of the research showcase.